Inside your cells, a tiny traffic system controls repair, cleanup, and communication, and when that system slows, age shows up as stiffness, memory lapses, and low energy. Scientists have just spotlighted one protein, AP2A1, that acts like a traffic director for cell cleanup, and early research suggests tweaking it could restore more youthful cellular function. That matters. Clearer cellular traffic can mean better muscle maintenance, sharper thinking, and less inflammation, the everyday changes that preserve independence. In this video, first I'll explain, in plain language, what AP2A1 does, why researchers think it could reverse key signs of aging, and what the lab results actually show so far. You'll hear how this discovery connects to real-world outcomes for people 60 and over, what treatments might look like down the road, and how lifestyle steps may support these advances today. No hype, just clear facts, practical context, and what to ask your doctor if you're curious. Stay with me to separate promise from possibility, learn the next steps in clinical research, and discover simple actions you can take now while science moves forward. 1. The reality of aging, why zombie cells accumulate as we grow older. Cells that stop dividing but refuse to die, often called senescent or zombie cells, accumulate in many tissues as we get older. They enter this state after significant stress, repeated DNA damage, critically short telomeres, metabolic strain, or chronic oxidative stress can all push a cell into senescence. Once there, the cell halts replication, becomes resistant to normal cell death signals, and begins to secrete a cocktail of inflammatory molecules and enzymes known as the senescence-associated secretory phenotype, or SASP. This secretory mix can degrade local tissue structure and signal neighboring healthy cells into trouble. The body usually keeps the number of these cells low by clearing them with immune cells such as natural killer cells and macrophages. With advancing age, however, two things happen at once: more cells are driven into senescence, and immune surveillance that removes them becomes less efficient. Factors like reduced immune cell activity, chronic low-grade inflammation, and impaired tissue repair all blunt clearance. As a result, senescent cells linger longer than they should, and their SASP turns short-term protective responses into ongoing damage. Visible examples help make this real. In sun-exposed skin, senescent fibroblasts secrete enzymes that break down collagen, contributing to thinning skin and wrinkles. In joints, senescent chondrocyte and supporting cells increase inflammatory signals that worsen cartilage breakdown and stiffness, feeding into arthritis symptoms. Fat tissue with a higher burden of senescent adipocytes produces more inflammatory molecules that interfere with insulin signaling and metabolic health. Blood vessel linings harboring senescent endothelial cells become more prone to stiffness and plaque formation, contributing to reduced circulation and cardiovascular strain. There are practical ways to reduce the pace of accumulation and lessen the harm these cells cause. Regular moderate exercise has been shown to improve immune function and lower markers of inflammation, helping the body clear or tolerate senescent cells better. Maintaining a healthy body weight and good blood sugar control reduces chronic metabolic stress that fuels senescence in fat and other tissues. Nutritional choices that emphasize whole, plant-forward foods and limit processed sugars can lower systemic inflammation. Sleep, stress management, and staying socially and mentally active support immune resilience as well. On the therapeutic side, research is exploring targeted strategies to remove or silence senescent cells. Some experimental drugs called senolytics selectively clear senescent cells, while xenomorphics aim to quiet the harmful SASP without killing the cells. Early human studies and ongoing clinical trials are investigating these approaches for conditions like pulmonary fibrosis, osteoarthritis, and age-related frailty, though they are not yet standard therapy and need medical oversight. Complementary measures that boost immune function and improve cellular cleanup, such as exercise, vaccination where appropriate, and careful management of chronic conditions, fit naturally alongside emerging treatments. You can take concrete steps now. Talk with your healthcare provider about lifestyle changes that reduce inflammation and protect immune health. Ask whether any clinical trials or specialist programs focused on senescence-related therapies are appropriate for you. Small, consistent choices, daily walks, balanced meals, good sleep, and control of blood pressure and blood sugar translate into fewer triggers for cells to turn senescent and a stronger ability to clear them. Stay curious and proactive. The growing science around senescent cells offers real reasons for hope, especially when combined with practical, everyday habits that support healthy aging. 2. Osaka University's breakthrough, the AP2A1 protein as a key to cellular youth. A steady slowdown in how cells sort and recycle their parts is one of the quieter drivers of aging. Researchers know that when membranes, receptors, and damaged proteins stop being trafficked correctly inside cells, tissues lose resilience and inflammation rises. This decline in cellular housekeeping contributes to frailty, slower wound healing, and the low-grade inflammation many seniors experience. Osaka University researchers reported compelling evidence that the AP2A1 protein, the alpha subunit of the AP2 adapter complex used in clathrin-mediated endocytosis, plays a central role in maintaining that housekeeping. In laboratory studies, cells with healthier AP2A1 activity showed improved uptake and recycling of surface receptors and a clearer removal pathway for damaged proteins. The team's findings suggest that restoring or supporting AP2A1 function can reverse some cellular features commonly associated with aging, such as decreased receptor responsiveness and accumulation of cellular debris. On a molecular level, AP2A1 helps select which membrane proteins are packaged into clathrin-coated vesicles for internalization. That process is vital for regulating how cells respond to hormones, growth factors, and stress signals. When AP2A1 function declines, receptors linger improperly on the surface or become trapped, signaling goes awry, and cellular waste builds up. The Osaka work connects these trafficking failures to markers of cellular senescence and an increased secretion of inflammatory molecules, a pattern that many scientists link to age-related tissue decline. For example, improved endocytic sorting can restore signaling for insulin or growth factor pathways, helping cells react more appropriately to repair cues. Translating this discovery into practical options will take careful steps, but the pathway is clear. Potential approaches under consideration include small molecules that stabilize AP2A1 interactions, gene-based methods to boost its expression in targeted tissues, and combination therapies that pair trafficking support with existing regenerative strategies. Seniors should not expect immediate treatments yet, but the research points to measurable biomarkers, like improved receptor trafficking or reduced senescence markers in blood samples, that could guide future trials. Meanwhile, evidence-based lifestyle measures, regular physical activity, balanced nutrition that supports protein turnover, and management of chronic inflammation remain sensible ways to help cellular maintenance while science advances. If this discovery feels personally relevant, consider 2 practical next steps. Talk with your primary care provider about participating in reputable clinical research and ask whether trials related to cellular repair pathways are recruiting. Many universities maintain registries and patient advisory groups. Stay informed through trusted medical centers, and prioritize clinician-guided lifestyle practices that support cellular housekeeping while researchers move AP2A1-based strategies toward safe, tested therapies. 3. Transforming old cells. How suppressing AP2A1 can restore vitality in later life. AP2A1 is the gene that helps build a key component of the cell's machinery for bringing things in from the surface, the AP2 adapter that drives clathrin-mediated endocytosis. Cells rely on that process to recycle receptors, clear damaged proteins, and regulate signaling. As we age, many cells become less efficient at those tasks, receptor recycling slows, waste builds up, mitochondria falter, and a low-grade inflammatory program called the senescence-associated secretory phenotype, SASP, can take hold. These changes underlie common problems seniors notice: slower wound healing, reduced muscle repair, and increased inflammation that saps energy. Emerging research shows that carefully reducing activity of AP2A1 in old cells can reverse some of those dysfunctions in laboratory settings. When endocytic balance is shifted, receptor populations on the cell surface change in ways that can restore useful signaling rather than perpetuate stress signals. That can mean better growth factor responsiveness in muscle stem cells, improved phagocytosis and pathogen clearance by immune cells, and more stable neurotransmitter receptor trafficking in neurons. In short, shifting AP2A1 activity may help aged cells move from a defensive, worn-down state back toward a more youthful, active one. Laboratory approaches used to achieve this include targeted genetic techniques, such as RNA interference or CRISPR interference, and developing small molecules or antisense therapies that dampen AP2A1 expression or function. Delivery matters. Researchers focus on tissue-targeted methods, nanoparticles, ligand-directed carriers, or local administration, so other essential endocytic processes aren't broadly disrupted. Transient modulation is another safety strategy, short-term suppression to reset cellular systems without permanently blocking a protein that cells need for many normal tasks. Rigorous preclinical testing looks for unintended effects on hormone signaling, neurotransmission, and immune surveillance, because those are real concerns whenever a fundamental pathway like endocytosis is altered. Translating those cellular gains into practical benefits for people involves steps many readers can relate to. Restoring better receptor and mitochondrial function in muscle stem cells could improve recovery after a fall or boost the benefits you get from a walking program. Calmer, less inflammatory tissue environments tend to heal faster and produce less chronic joint stiffness. For cognition, improved receptor trafficking and mitochondrial health can support attention and memory, particularly when combined with mental activity and good sleep. These are not guarantees. They are plausible outcomes being tested, and the most likely route to safe, effective therapies will combine precise molecular tools with lifestyle measures that seniors already use to stay healthy. If this line of research interests you, start by talking with your healthcare provider about emerging trials and what biomarkers or functional tests to monitor, things like measures of inflammation, physical performance tests, and cognitive screening. Consider tracking practical indicators that matter in daily life: walking endurance, hand grip strength, frequency of infections, and wound healing time. Stay curious but cautious. Therapies that modulate AP2A1 are a promising frontier rather than an established cure. Ask about clinical studies that match your health profile, and maintain habits that support cellular resilience while science works on the rest. 4. The role of IU1, helping the mature body clear out damaged proteins more effectively. Our cells rely on the ubiquitin proteasome system to tag and remove damaged or misfolded proteins, and that cleanup service tends to slow down as we age. When the proteasome lags, damaged proteins build up in cells, contributing to stiffness, loss of muscle quality, and increased risk of neurodegenerative problems. This decline in proteostasis is a well-recognized challenge for people over 60, because long-lived cells, like neurons and muscle fibers, are particularly sensitive to accumulating protein waste. A promising laboratory finding centers on IU1, a small research molecule that targets a ubiquitinating enzyme called USP14. Cells normally attach ubiquitin tags to proteins destined for destruction, and USP14 can remove those tags, giving a second chance to proteins that should be cleared. By inhibiting USP14, IU1 lets more tagged proteins proceed into the proteasome for final breakdown. In preclinical studies, this has translated into faster proteasomal removal of several aggregation-prone proteins seen in aging tissues. In simpler terms, IU1 acts like a helpful traffic cop at the entrance to the cell's recycling center. By preventing the undoing of the destruction tag, the proteasome gets a steadier stream of material to process. Laboratory experiments in cultured cells and animal models reported improved clearance of misfolded proteins, including some forms of tau and TTP43 that are linked to neurodegenerative disease, along with reductions in cellular stress markers. These results are encouraging because they show that proteasome activity can be boosted pharmacologically, not just by lifestyle changes. Practical benefits for older adults could include slower accumulation of toxic protein clumps and better cellular maintenance, but it's important to be clear, IU1 itself remains a research compound, and its safety and effects in people are not established. There are also scientific caveats. USP14 touches many cellular pathways, so broad inhibition can have unintended consequences. That's why researchers are working on more selective compounds, delivery methods that target vulnerable tissues, and combining proteasome-enhancing approaches with therapies that stimulate complementary cleanup systems like autophagy. If you're curious and proactive about healthy aging, the most useful steps today are practical and evidence-based. Keep physically active, prioritize good sleep, control blood sugar and inflammation, and discuss with your clinician whether participating in clinical research is appropriate. Stay informed about trials that aim to translate IU1-like strategies into safe therapies. Small daily choices support the same cellular housekeeping that experimental drugs seek to enhance. Together they form a realistic, hopeful path toward clearer cellular housekeeping as we age. 5. Visible results: how laboratory tests showed old cells regaining youthful functions. Cells taken from older donors often show clear, measurable declines in the things that keep our tissues resilient: slower division, weaker mitochondria, persistent inflammatory signals, and a tendency to stick in a senescent state. Scientists use a panel of laboratory tests to quantify these changes, because visible cellular behavior helps tell the story of biological age more reliably than appearance alone. When a targeted change in cellular machinery produces repeatable shifts across those tests, the result is more than promising. It becomes visible evidence that some aspects of cellular aging are reversible. Researchers investigating AP2A1 focused on exactly those measurable features. In cell cultures where AP2A1 activity was increased or normalized, laboratory assays showed older cells recovering traits typical of younger cells. Tests commonly used by aging researchers registered shifts, fewer cells stained for senescence-associated beta-galactosidase, higher rates of cell cycle markers such as Ki67, and improved mitochondrial function as seen in dye-based membrane potential assays. Those are not abstract labels, they correspond to cells that are dividing again, producing energy more reliably, and less likely to secrete chronic inflammatory factors. Microscopy and functional uptake assays made the change obvious in images and plots. Under the microscope, actin structure and membrane dynamics looked more organized, and clathrin-mediated endocytosis, often measured by fluorescent transferrin uptake, returned toward levels typical of younger cells. Mitochondrial assays using common dyes showed increased membrane potential and steadier ATP production curves. Meanwhile, secreted proteins that form the senescence-associated secretory phenotype, SASP, were reduced on cytokine panels, indicating a quieter, less inflammatory cellular environment. Epigenetic assessments often used by labs, such as shifts in DNA methylation patterns measured by established methylation clocks, also trended toward a younger profile in many samples, reinforcing the visual and biochemical findings. Putting these laboratory signals together gives a coherent picture that matters for everyday life. Cells that regain robust endocytosis and mitochondrial health are better at clearing damaged proteins, responding to stress, and supporting tissue repair. Lower SASP output means less chronic inflammation at the cellular level, which is linked to common age-related complaints like stiffness and slower healing. These laboratory-visible improvements do not promise instant rejuvenation, but they do suggest mechanisms through which function in tissues, skin, muscle, immune cells, could be supported or improved over time. Talk with your healthcare team about what the science means for you. Follow credible trials and updates from research centers, and consider lifestyle measures known to support cellular health, balanced nutrition, regular physical activity, quality sleep and stress management, while researchers continue to translate laboratory findings into safe, practical therapies. If you're interested in participating in research, ask clinicians about reputable clinical trials. Being informed and involved helps bring these visible laboratory advances closer to meaningful, real-world benefits. 6. A new era of longevity. Why 60+ is becoming the new prime of life. Cells in older adults often show slower protein turnover, impaired receptor recycling, and an accumulation of damaged cellular components that together underlie fatigue, weakened immunity, and reduced tissue repair. These are not abstract laboratory problems, they help explain why recovery from illness can take longer and why chronic conditions become more common after 60. The good news is that molecular research is revealing specific mechanisms behind these changes, creating clear targets for interventions that go beyond symptom management. One promising avenue involves proteins that control how cells traffic receptors and cargo, such as AP2A1, a subunit of the AP2 adapter complex responsible for clathrin-mediated endocytosis. AP2A1 helps determine which receptors are pulled into the cell and whether they are recycled back to the surface or sent for degradation. When that sorting machinery works well, cells maintain sensitivity to hormones, clear signaling debris, and regulate inflammation more effectively. When it falters, signaling pathways can become sluggish or dysregulated, and that can show up as slower gait speed, diminished vaccine responses, or longer recovery from injury. Translating that biology into tangible benefits for people over 60 is already underway in concept and early-stage research. For example, enhancing receptor recycling in immune cells could improve how seniors respond to vaccines and infections, while stabilizing neurotransmitter receptor trafficking may support cognitive resilience. In muscle and bone, better turnover of growth factor receptors and surface proteins can help preserve strength and balance. These are realistic pathways. Laboratory models and cellular studies indicate that improving intracellular trafficking restores more youthful patterns of signaling and repair. It's not magic, it's targeted repair of a cellular housekeeping system that has wide downstream effects. Practical integration of this science into daily life is straightforward and empowering. Continue or begin strength training to boost muscle protein synthesis and receptor responsiveness. Prioritize sleep and manage chronic inflammation through diet, medication review, and regular cardiovascular activity. These habits support protein clearance systems throughout the body. Ask your clinician about biomarker testing and whether local hospitals or universities are running clinical trials that target cellular trafficking or proteostasis pathways. If you take multiple medications, review them with a pharmacist. Sometimes simplifying drug regimens reduces stress on cellular clearance systems. Take a proactive step today. Talk with your healthcare team about emerging therapies and trials that focus on cellular maintenance pathways, and pair those possibilities with proven lifestyle choices that support protein health. Science like the work around AP2A1 is expanding the toolbox for resilience and function, so the decisions you make now, both small daily habits and informed medical conversations, can help make the years ahead some of the most active and fulfilling of your life. Aging has quietly changed the way your body moves, remembers, and recovers, and that can feel like losing parts of your independence. The good news is the AP2A1 protein discovery points to a realistic path forward. Early research shows restoring AP2A1 helps clear cellular clutter, improve cell-to-cell communication, and may reverse key signs of cellular aging in laboratory studies. Summary of key points: AP2A1 plays a role in healthy protein traffic inside cells. Boosting its function has led to rejuvenation effects in preclinical models. This is promising but still experimental, and human trials are the next step. You're not being promised a miracle, you're seeing a hopeful scientific advance that could translate into safer, targeted therapies down the line. If this feels relevant to you, start simple, talk with your doctor about ongoing trials, maintain activities that support cellular health, balance nutrition, gentle movement, sleep, and stress management, and stay informed about developments. Your lived experience and questions matter. Researchers need voices like yours in clinical studies. If this video helped you feel more hopeful, please like, subscribe, and share with friends who care about staying active and independent. Tell us in the comments what matters most to you about aging well, and we'll cover it. Coming up next, a closer look at clinical trials, safety considerations, and practical steps seniors can take today to support healthy cells. Stay curious, stay empowered, we're on this journey together.